with the hammer, okay? So I will chair this meeting then, and I give everybody a few minutes to come in. So then in the meanwhile, so Josh, I guess you had a look at the website of this Jolly meeting series, and you see the names from the Netherlands that you know already from the conferences.
I did.
So it started as a quite informal gathering organized by Peter, saying, guys, we are doing the same thing. Why don't we do it together?
Sure.
And then we started to talk to each other domestically, writing project proposals, launching a series with special speakers, and here we are. Yeah, it's quite an interest. So at least 4 partners from the Netherlands, 4 cities.
Great. Yeah, looking forward to catching up with some old friends here and make some new ones.
Yes, okay. Too bad we cannot do it in real life, but probably in real life it wouldn't be so easy to invite each other and then,
True.
get yourself over here. But I think I said it before, so you have a rain check whenever you are in the Netherlands. I would be really happy to sit down and drink those beers that we missed the last 2 years.
Yes, exactly. The numbers are increasing. Yes.
Yes. Yeah. So it sounds like a lovely evening, right?
Agreed. And I would extend the same invitation to you and your group. If anyone passes through the Northeast U.S., please come and stop by the University of Vermont. It's a beautiful part of the world. Although it is still snowing here. We seem to be stuck in the winter that will never end. But if you like the snow, this is the right place to visit.
Oh, thank you. On behalf of everyone, you are in danger, obviously, considering the gecko in Boston. But
yes, right. Yep. Yeah, let me know if a side trip is possible. We'd love to host you.
Oh, nice. Okay, so let me welcome everyone to today's gathering of the Jolly session, the joint lecture series on evolutionary. And today we have a very special speaker from the United States who is doing really exciting things in evolutionary computing / evolutionary robotics / artificial life. So this is the moment when I say he doesn't need introduction, but I will introduce him a little bit. And I will jump a period of 15 years at least. So the first time I heard about Josh was when he was working together with Ralph Pfeiffer. I wrote this seminal book, this lovely book, How the Body Shapes the Way We Think, which I love to paraphrase as How the Body Shapes the Brain. And from that moment on, he is doing really cool things, and partly because he is interested in how the body and the brain influence each other on the environmental circumstances and on the evolutionary pressures. So this is an exciting thing because it somehow gives a promise to disclose something about our own origins, the origin of life on Earth and the origin of human intelligence. So that's where it all started, and then he had a couple of very interesting papers in very interesting journals, which I will not sum up, but I invite you to go and look up his webpage or the Google Scholar list of his publications. But let me jump 15 years, or 14 years to when he did something completely incredible. He was fed up with the criticisms that these little walking sponges that were to simulate some multicellular creatures, would not be interesting for studying in simulation because they don't have a real world equivalent. So he took up the challenge and liaised with biologists. And then last year he came up with a solution. He came up with a paper that showed a physical incarnation of those creatures based on organic biological matter. So that's what became very famous as under the name of xenobots. So today he will give us an overview of everything, or a lot at least, of his career and of his works, and obviously also touching on the xenobots. So Josh, I give you the floor. Thank you again for being here.
Thank you so much, Gus, and thanks to all of you for the invitation to come and present some of our work here. So just a couple of housekeeping things before we get started. I can see chat on my secondary monitor, so please feel free. It would be great for this to be a discussion rather than a lecture. So if you have any questions, please type it into chat, and I'll try and answer in situ. And I'll also make sure to leave some time at the end for some questions and answers. So as Gus promised, I'm going to give you an overview today on sort of my 15-year journey from rigid robots to soft robots, and most recently in the last 2 years, biological robots. And I want to characterize this journey for you as a walk through Robospace. So in evolutionary biology, there is this theoretical construct known as morphospace, the space of all possible organisms that currently exist, have existed, or don't exist at all, where every point in this theoretical space corresponds to a distinct biological species. We can borrow that same idea to organize where we've been as a robotics community and think of that as Robospace. And obviously, this is kind of an imaginary high-dimensional space, so you can choose your axes as you wish. As Gus mentioned, starting with my PhD work with Rolf Pfeiffer, I've focused on this idea of embodied cognition, or how robot bodies can facilitate learning intelligence rather than obstruct it. So one of my axes on Robospace here is embodiment, walking from the front of this cube towards the back. We're visiting increasingly embodied machines, starting with naked neural networks and ending with Asimo or soft robots or biological robots. The horizontal position of each point here, which represents a given machine, is how good it is at whatever we want it to do. And the higher that point is, the more general purpose that machine is. Given those three axes, we can sort of think about where AI and robotics has been so far, which is traveling from the left to the right side of this cube. Thanks to the deep learning revolution and the big data revolution, we can produce very, very accurate and performant specialized machines. We have a harder time as a community moving up in this space, creating more and more general purpose machines. And it gets harder and harder to produce useful and accurate and safe machines as we move from the front of this cube towards the back. So as a community, we've explored some of this space, but there are vast parts of Robospace that we have not visited at all. So what I'd like to do today over the next 45 minutes or so is just touch on some of the places that I've been with my colleagues and now the students in my group. And I'm going to focus on some points in Robospace here that contain non-intuitive solutions. These are machines that look and act in ways that, at least for us, was not obvious beforehand. Like many of you, the vehicle we use to travel through Robospace is evolutionary computation. I'm assuming that most of you are familiar with evolutionary computation. I'm not going to talk about EC itself. I don't really consider myself an expert in EC. We sort of use and adapt EC methods slightly to try and direct where we go in Robospace. And again, we try and direct where we're going into increasingly non-intuitive regions of Robospace. Okay. One of the things I think that's holding us up as an AI and a robotics community, part of the reason why we're having a hard time exploring the space, creating useful, competent, general purpose embodied machines, is that at least in robotics, up until recently, our machines have been conceptually hollow. What do I mean by that? Traditional robots, especially rigid robots, they're built from inert materials, steel, plastic, ceramics. An individual component of a robot is not itself intelligent, and that causes a lot of difficulties. Our current robots don't have a rich inner life, so it makes it very difficult for them to grapple with the richness of their external life, the way in which they can interact with their environment. I like to call on the 17th century, the 18th century philosopher David Hume here. He introspected about a century after Rene Descartes, and Descartes had seemed to have found this constant self. There was this nugget of unchanging res cogitans inside that was the self. Hume came to the opposite conclusion. When he looked inside, when he mentally introspect, he didn't see anything that was constant. He saw a constant flux instead. In Hume's own words, his internal images and feelings seem to be in this perpetual flux and movement. He could not find this unchanging self that Descartes purportedly had found a century earlier. So, as human beings, subjectively, our mental life is constantly in flux. And of course, our physical life, our physiological self, is also constantly in flux. Our bodies and brains are changing all the time at spatial scales ranging from the nanometer all the way up to the whole body. And these adaptive internal or interoceptive processes are unfolding over time scales from the nanosecond to decades and centuries and millennia in some plant species. Increasingly in my group, we try and demonstrate or argue that ability to constantly deal with internal change pre-trains organisms to deal with external change. So I'm deliberately using a term from machine learning, which is pre-training. Why are organisms so good at dealing with novel environments, even environments they've never seen before? Sometimes they do it with few shot, sometimes they do it with zero shot learning. I would argue it's because their cells, their organs, their tissues, their whole bodies have dealt with and constantly deal with internal change. So I'm going to walk you through some work from my group where we've gone from rigid to soft to biological robots. And the thread that ties these three groups of machines together is increasing opportunity for internal change. Rigid robots are incapable of internal change. Soft robotics, thanks to material science advances, are able to change their internal structure and dynamics in more ways. And finally, in biological robots, if we're going to build robots completely from living cells themselves are constantly changing. They constantly change how they communicate with their neighbors. They are, by definition, machines that are constantly in internal flux. So let me start with rigid robots. This is some work conducted with Hod Lipson and Viktor Zykov back in 2006. I'm not going to go into the details of this experiment, but what we showed here is a robot that is able to deal somewhat with internal change. It's unanticipated and unexpected internal change, which is physical damage. So a lot of what we've done in my group over the years is to torture robots, not because we don't like robots, but because it's a great example of trying to grapple with unexpected internal change, which is arguably also external change. What you see on the left here is our evil starfish. This was the nickname given to this robot by an undergraduate working in the lab. The evil starfish here has learned how to walk on its own by developing a self-model, which you can't see in this video. It internally trains a way of walking using that self-model, which allows it to walk. When it becomes damaged, it's no longer able to walk. And in order to learn to walk again, it creates a new self-model, uses that self-model to train a new control policy that compensates for the injury, and off it goes. Of course, what's happening here is that our robot is hobbled in two different ways. It's hobbled literally because it's missing half of one of its legs, but it's also hobbled in a fundamental sense that when something surprising happens, like losing part of a leg, rigid robots have only one option open to them, which is to adapt their control policy, the way in which they move. I'm going to switch now from rigid robots to soft robots. This is much more recent work with Rebecca Kramer-Bottiglio's lab at Yale and Mike Levin's group at Tufts. In this case, we simulated some soft robots, and like you just saw, we lop off one or a few of their legs and look at how they adapt. Like rigid robots, one option available to soft robots is to adapt their control policy, how they move. And even if you lose half your body as a soft robot, at least in silico, you can still recover some functionality. However, because this robot is soft, there are other opportunities available to it. So here you see an undamaged four-legged soft robot. It's evolved a controller that allows it to move perfectly fine. And as you're going to see in a moment, we're going to lop off all four of its legs. And in this case, it does not adapt its control policy. It adapts its body. It regenerates the lost leg, and it's actually running on board. It's running on board the original undamaged control policy. So it's adapted its body rather than its brain in this case. I promised you that we'd visit some non-intuitive parts of Robospace. Here's another example from exactly the same experiment. In this case, we're going to chop off two of its legs. And you'll notice that instead of regenerating the lost legs, it instead retracts the surviving two legs and reverts from legged locomotion to peristaltic motion, even though it's running the same pre-damaged control policy. It has not adapted its control policy. For us, this was a big surprise. We were expecting when we designed this experiment that most of these injuries would cause the robot to change its body to regenerate the lost limbs, but it came up with regeneration as one strategy, but a bunch of different strategies as well. Goose mentioned Goose has a question. Go ahead, Goose.
You encourage us to interrupt you, so I do, okay?
Please.
Yes, this is a very convincing demonstration, but the question is, wouldn't a rigid robot could show a similar response by, if it had joints, just, bending differently purely by gravity and having the same kind of effect, growing a leg where there was no leg before?
Possible. Yeah, it's possible that if you have a rigid robot with enough mechanical degrees of freedom, it would be possible to approximate some of the strategies you see here, like regenerating a leg or retracting or hiding a leg. But it's usually difficult, and you need large numbers of mechanical degrees of freedom to do that. There are also large numbers of mechanical degrees of freedom in the soft robots that you see here. Every individual voxel could, in theory, be actuated differently. So you're right. In theory, it's possible. I think in practice, however, given the mechanical construction of soft robots, it's easier for evolution or learning methods to find some of these non-obvious solutions. Yep, good question. Yeah, please do feel free to interrupt it at any time. Okay, so that was just an example again of sort of the opportunity that soft robots afford through shape change. So this is one form of interoceptive or internal change that's possible. Here's another example where the internal change you're going to see is not at the spatial scale, not just at the spatial scale, not just shape change, but I'm going to show you internal adaptation occurring across two different time scales. We have the fast time scale oscillation of the control policy that you see here, but you'll notice that there is a slower change occurring in the body. The robot is slowly changing its shape as it's actuating or trying to oscillate and move. By combining these two time scales together, you can create soft robots which exhibit the Baldwin effect, and I'll show you visually what the Baldwin effect is in a moment. Okay. Turns out that a robot that changes its shape slowly can sometimes partway through its lifetime discover a new behavior, like in this case, rolling. This robot is running one control policy, but also changing its shape. This rolling behavior is a very rare point in the behavioral space for this particular robot. In English, we call it a needle in the haystack. It's a really hard thing to find. But if you have robots that are changing their shape, they are not necessarily a point in behavioral space, but they are drawing or they're moving through a small area or a small volume in behavioral space that allows some of these robots sometimes to accidentally discover rare behaviors. In this particular case, I'll show you an evolutionary, this is one evolutionary run. Here's an ancestor moving at random that does not discover this needle in the haystack. Its descendant discovers walking, which is a common strategy in this behavioral space. Its descendant, as I just showed you, accidentally discovers rolling towards the end of its lifetime. Its descendant, in turn, earlyifies. There are mutations that cause it to change shape so that it discovers rolling earlier in its lifetime. And its descendant genetically assimilates this needle in the haystack, this rare, useful behavior. This is an embodied version of what's known as the Baldwin effect in evolutionary biology. So another advantage of incorporating internal change, a robot that changes shape slowly while actuating or behaving or locomoting quickly. Okay, so I just showed you stacking together temporal change internally. This is some more recent work, again with Rebecca Kramer-Bottiglio's group at Yale, where now we're going to stack structure together. We're going to stack bodies together structurally rather than temporally. These are, as you can see, fractal robots. The question we asked here is, can you evolve a robot like the one you see on the left that exhibits some desired behavior, which is movement from left to right? And if you do, and you stack that robot self-similarly, like you see in the middle and the right-hand panel, does that structural self-similarity confer behavioral self-similarity? Meaning that the bot, or the bot made of bots, or the bot made of bots made of bots, that they all exhibit the same behavior, but at different scales. Here's an example of a particular fractal robot that does. That base shape or that base structure, which also dictates how to put together copies. In this case, structural self-similarity does result in behavioral self-similarity. Turns out that that's not always the case. There are certain shapes and behaviors that if you combine them self-similarly, you have structural self-similarity, but you do not necessarily get functional or behavioral self-similarity. So this is a sub-region of Robospace, which is the space of all possible fractal robots. And we kind of stumbled across this sort of by accident. And it suggests these new kinds of research questions that at least are new to me in robotics. So again, the body shapes the way that we think about AI, robotics, embodied cognition. Organisms are fractal in every possible sense, right? We are stacked together structurally and functionally self-similarly. It's another kind of internal structure or internal dynamics that is usually missing from robotics. I think there's a lot of interesting work that can be done here when we think about fractal structure and dynamics interoceptively inside the machine. Okay. As Gus mentioned, usually we hide in a virtual space. This was one of the rare cases where we actually went to the effort, or at least our Yale colleagues went to the effort of building a physical version of our fractal robot. It doesn't quite behave functionally self-similarly yet, but we're getting there. Okay, I just want to pause now before I switch gears. I've showed you some rigid robots and some soft robots. And here's an example of them in the top row. We worked on all sorts of other kinds of machines in the past and recently. In the bottom row, in the bottom center, those are the biological robots or the xenobots that I'm going to talk about in a moment. But before I do, I want to take a little bit of a digression and talk a little bit about what you see in the bottom right, which is not strictly a robot. These are metamaterials. And I think metamaterials is kind of a sister discipline to robotics and soft robotics that has a lot of promise for helping us achieve embodied intelligence. So for those that are not familiar with metamaterials, here are 3 simulated granular metamaterials. As the name implies, granular metamaterials are metamaterials built from grains. A metamaterial is an artificially designed material that has bulk properties. So stiffness it acts as a material in ways that you typically don't see in nature. Common example of a mechanical metamaterial is if you take a natural material and you compress it from the top and the bottom, it will expand to the left and the right. You can design mechanical metamaterials that when you compress from the top and the bottom, it also compresses from the sides. And you can design these metamaterials to exhibit bulk properties more and more orthogonal to the bulk properties you see in natural materials. Not too much interesting stuff going on here. What you see in these 3 different metamaterials, both the simulated and the physical version, is we're applying a steady oscillation from the left-hand side. And you'll notice that individual grains inside the material, some of them amplify the amplitude of that oscillation and some suppress it. And if you look carefully, it might be difficult to see in the video, there is a nonlinear relationship. So you can design, even in these very simple metamaterials, you can get some very interesting vibrational properties, which holds a lot of promise for soft robotics. If you have vibrations passing through the body of a robot, and those vibrations are being caused by external events or internal events, those oscillations can be exploited to direct force. So you can create actuators. They can be used to propagate energy through the system. They can be used for communication. They can be used as a form of sensing. This is another aspect of soft robotics that is very interesting. The kinds of embodied cognition we're starting to see emerging in soft robotics is a blurring between the distinction of actuation, control, sensing, energy storage, compliance, and so on. Again, in traditional robotics and rigid robotics, we typically think of those as distinct components, but you can actually mix them in soft robots, and metamaterials provide a particularly interesting way of mixing actuation, power, sensing. And what I'd like to show you just very briefly is computation as well. Here's a simulated 2D metamaterial. There are 2 grains that we're interested in this case, the green one and the blue one. You'll notice that we are oscillating the green one. We are holding the blue one clamped. And you'll notice that the red one starts to exhibit some of this oscillation. You'll notice that there's a mixture of dark gray and light gray circles here. The light gray are soft particles and the dark gray are stiff particles. It turns out that if you evolve these metamaterials, so you create a population of materials that mix soft and rigid components, you can then evolve these metamaterials to act computationally. So in this case, this is some work by my PhD student, Atusa Parsa. And what Atusa did in this case was to create a fitness function that rewards metamaterials that act increasingly like an AND gate. So in this case, you can evaluate this simulated metamaterial 4 times, vibrating neither one or both of the green and blue particles, and observing vibrations arriving at the red particle. And it turns out there is a combination of rigid and soft particles. It's this combination that you see here that will produce or allow the vibration to reach the red particle only when both the green and the blue particle are being vibrated. So we're representing bits as oscillations here, and we can again evolve these materials to act increasingly like an AND gate. They have more and more AND-ness at a chosen low frequency. We choose that frequency for particular reasons. I won't get into that today. I don't have time. An interesting thing that you can do, and Atusa did this, and we're reporting this in an upcoming Gecko paper this summer, you can extend this into a multi-objective optimization exercise where now you're evolving populations of metamaterials and you're evaluating them 8 times. The first 4 times you want to see how much it acts like an AND gate, and the second 4 times you want to see how much it acts like an exclusive OR gate, but at a higher frequency. I just showed you that at one end of the Pareto front, you get materials that act very much like an AND gate and act terribly as an exclusive OR gate. At the other end of the Pareto front, you get, in the same evolving population, you get metamaterials that act very much like exclusive OR at high frequencies. What you get at the knee in the Pareto front is something that's particularly interesting. You get a material that can act as an AND gate and an exclusive OR gate, and it can act as both gates at the same place at the same time. So if we multiplex the low and the high frequencies that we're supplying, we are simultaneously supplying different combinations of zeros and ones at low frequency and high frequency. And if we take a fast Fourier transform, we measure the amount of power of the low and the high frequency at the red particle. It turns out that this particular metamaterial that you see here, the simulated metamaterial, the red particle acts simultaneously as an AND gate at low frequencies and an exclusive OR gate at high frequencies. So we refer to this as computational superposition. There's many things I think that's interesting about this. One of them is that this material is acting as is performing computational superposition without recourse to quantum effects. This is a particularly non-intuitive corner of Robospace. Again, this isn't necessarily a robot, but a lot of interesting opportunities start to arise when you think about trying to create robots built from metamaterials. This is related to the sister disciplines of physical computing, mechanical computing, physical reservoir computing, morphological computation. I think, again, this is just the tip of the iceberg. As materials improve, we as a community can take our evolutionary algorithms and push more and more into these non-intuitive corners of Robospace. Okay, last but not least, I will finish by introducing you to our biobots, or our xenobots as they become popularly known. I'll tell you a little bit about the origin story for the xenobots here. My biology colleagues Mike Levin and Doug Blackiston back in 2013 published this very interesting paper where they explanted eye precursor cells into the tail of a tadpole. And not only did that surgical procedure not kill the tadpole, they also inhibited the developmental expression of eyes on the front of the robot, the front of the tadpole. So they've surgically blinded this tadpole, but given it the opportunity to see by giving it eye precursor cells in its tail. So not only did this surgical procedure not kill this tadpole grew into a perfectly healthy adult frog. And during development, these eye precursor cells matured and developed into a functioning eye. That eye established a neural handshake with the developing spinal cord as well. So at the end, you have an adult frog with an eye on its back, and it was able to actually use this eye for phototaxis and food capture. There are a lot of other studies in the literature that show that biological organisms are particularly willing to absorb rearrangements of their components during development. And I think this is sort of the end game of this internal flux that David Hume mentioned back in the 18th century. Organisms are very good at dealing with constant change. All of us grew from a single cell, and we managed to stay alive during that process as we grew into trillions and trillions of cells. If we can manage that, being able to handle an ectopically implanted sense organ is sort of easy stakes by comparison, right? So how do we exploit that, that ability of living tissue to absorb rearrangement? This experiment and some others led us to ask the following question: is could we bring in an evolutionary algorithm to decide how to rearrange biological tissue? And could it search for particular rearrangements of biological tissue that result in a adult healthy organism that looks and acts as differently from the adult default phenotype as you would expect? So this particular tadpole grew into a frog adjacent adult phenotype. Looks and acts mostly like a frog, with the exception that it's got an eye in the wrong place. How far could we push that idea? So to study that, we started this project about 4 years ago now, and working with our microsurgeon colleague here, Doug Blackiston. Doug told us that he could relatively easy harvest two types of cells from early frog embryo, basically a frog egg. He could harvest stem cells that are fated to become a type of skin, and also myocardiac cells. So these are cells that spontaneously increase and decrease in volume. And if myocardiac cells arrange themselves during frog development into a frog heart, they spontaneously synchronize and pump blood in and out of the heart, very much like human myocardiocytes do. So we took those two cell types and we turned them over to our soft robotics simulator. And we told our physics engine here a small amount of biological detail about these two cell types, but there was a lot of biological detail we left out. You can see that the skin cells here became cyan voxels. These are passive soft voxels that can be pushed and pulled by forces acting on their surfaces. And the myocardiac cells became red-green voxels, which if you squint, you'll notice that they're increasing and decreasing in volume. We asked our microsurgeon colleague, Doug, what would happen if we put together frog myocardiac tissue not in the shape of a heart? Would they synchronize or not? Doug said they'll probably synchronize in certain conditions and not synchronize in other conditions. So taking a page from the sim to real literature, we were very conservative in that no matter how the evolutionary algorithm rearranged or composed simulated myocardiac tissue, it never synchronized. So we're giving our evolutionary algorithm here a very difficult task, which is to build a reliable machine, build a simulated xenobot that moves as quickly as possible from left to right, produce reliable whole body behavior, but you have to produce it with unreliable parts. Any individual myocardiac cells, which are basically the pistons or the actuators of this machine, they don't synchronize. So one way to think about how this is so difficult is imagine you were tasked with building a boat, and you could build the shape of that boat however you wanted. And you could place into that boat as many human rowers as you want. And you can put those rowers wherever you want, but the rowers are not going to synchronize how they row the boat. What is the 3D geometry and where should you place the rowers so that you get a boat that goes straight as quickly as possible? It's a very difficult engineering task. This is obviously exactly where we want to try and use evolutionary algorithms. So the first step in this experiment was to verify that an evolutionary algorithm even could design such a machine. And I wouldn't be here today if the answer was no. Turns out the answer is yes. Here is a evolved simulated xenobot. You'll notice that there's some interesting 3D geometry here and some interesting distribution of these voxels. The 3D shape causes the mass distribution to kind of tip forward. And then the evolutionary algorithm has placed a lot of these pistons or these randomly increasing, decreasing volumetric voxels on the ventral or the lower surface. And you'll notice that it's put this the soft skin on the top. And if you look very carefully, you'll notice that the top part of the robot is kind of absorbing all the individual impulse forces coming from below. And in a way, it's kind of averaging or smoothing out all those indirect actions and somehow combining or averaging and smoothing and directing all of those forces to produce reliable, non-random forward motion. This is, I think, a great example of a morphological computation. It's kind of a soft robotics version of passive dynamic walking. So using some of the passive parts of your body to direct motion. The next step, obviously, is could Doug actually build a physical version of what we evolved? Here's an example of Doug building not actually that simulated xenobot. He's trying to build one of our four-legged soft bots that Doug saw early on when we started this project. What he's got here is a clump of just frog skin cells, so no myocardiac tissue here. So this is a xenosculpture, not a xenobot. It can't move. We're looking down through the microscope with Doug, and you can see that he's using some microsurgical tools here to scrape away some of the material. And he's actually got this Robosculpture flipped on its back, and he's removing some of the material to create these four legs. This clump of frog skin cells that you're looking at is about 1 millimeter across. So all of the xenobots are, in effect, micro robots. Doug then went to try and build a physical version of the evolved simulated xenobot that I just showed you, where he deposits a layer of frog skin cells and then a layer of frog myocardiac, then a layer of skin, layer of myocardiac, kind of building up like a sandwich and then scraping away excess material. So we have automated design. Thanks to our evolutionary algorithm, but very, very manual manufacture. We refer to this as artisanal robotics. Takes Doug about 4 hours to make 1 millimeter sized xenobot. So obviously a limiting factor of this combined approach at the moment is the manufacturing side. Turns out that for this particular design, we were able to cross the sim to real gap. You'll see that the physical xenobot looks similar, but not identical to its simulated counterpart. And it also doesn't move identically to its simulated counterpart. But more or less, we get more or less the same kind of behavior. To verify that this physical xenobot is moving because of its shape and distribution of material properties, we took this physical xenobot and we flipped it on its back. Here you see it's moving right side up, and in a moment you're going to see it on its back. And when we flip it on its back, it no longer moves. So this was evidence that it's not just spontaneously moving because xenobots like to move. It's moving because of the geometry and distribution of tissues discovered by the evolutionary algorithm. At the end of this first paper, we included a few videos basically showing, kind of for fun, we took a bunch of the xenobots that we had made and put them in the dish together and got some interesting collective behavior. Now remember, these xenobots were evolved for individual behavior. They're not evolved to do anything smart collectively, but kind of like Roombas or Rolf Pfeiffer's original Swiss bots. If you sprinkle very, very small glass beads into the dish, it's hard to see in the physical video, but they will collectively kind of push these beads into piles. Whether the physical xenobots are doing this, whether they sense and react to the beads or each other, is still an unknown question. The xenobot as a whole was not evolved to have a sense, think, act cycle, but individual cells themselves are very sophisticated robots. They have very sophisticated sensing and actuation capabilities. So it's possible that the xenobots themselves are sensing and reacting to their environment, even though they were not evolved to do so. So these observations from this collective behavior led us last year, I'm going to skip ahead a little bit, led us last year to study what would happen if instead of putting glass beads into the petri dish of these xenobots, what would happen if we sprinkle more dissociated frog skin cells? You can see these dissociated frog skin cells in the dish. Turns out that when these are taken from very early frog embryo, they're kind of sticky and they will glom onto one another. If you take a xenobot and put it in the dish, it again pushes these objects, these frog cells, into piles. And certain xenobots will create bigger piles than others. Turns out that if you create a large enough pile of individual frog cells that stick to one another, they will spontaneously grow cilia. Cilia are very, very small microscopic hairs that will spontaneously beat like flexible oars. And what you're seeing here is obviously a zoomed in xenobot that is now not walking, but swimming. All of the cilia on the surface of this bot are inducing flows around the robot that allows it to swim and very lightly touch or float along the bottom of the dish. The conditions under which a group of frog cells will grow cilia is still not very well understood. For our purposes, Doug, the microsurgeon, told us that what he had seen under the microscope is that a pile of frog cells with more than 50 cells in it will grow cilia. Piles with less than 50 cells doesn't grow cilia. We took that detail and we built it into another simulation and another evolutionary algorithm, which I'm going to walk you through. Now. So we started with this assumption that if we put a number of xenobots into the dish in simulation and allow them to push some of these simulated frog cells into piles, we could create an evolutionary algorithm that selects for a xenobot swarm to produce larger and larger piles. In this case, this is a swarm taken from generation 0 of the evolutionary algorithm. So now we have not a population of simulated xenobots. We have a population of simulated xenobot swarms. This particular swarm members have this spherical shape. They collectively make a bunch of small piles. And the pile that forms middle right here, this is 74% of the way to having 50 cells in it. So whatever that is, 74% of 50. So this particular swarm gets a fitness value of 0.74. A few generations later, this particular swarm evolves. In this case, the evolutionary algorithm is still designing the 3D geometry of our xenobots, and it turns out that for whatever reason, the swarm of toroids or donuts happens to produce larger piles. And when this video starts again, you'll see the initial swarm. The initial swarm makes a relatively large pile, upper left here, and that pile contains 50 cells. And so in reality, that pile of 50 cells would grow cilia and start to move. So we simulate that here. And we have 9 parent xenobots that collectively produce 1 child xenobot. They have managed to kinematically self-replicate. They have replicated by kinematically or by moving themselves and moving these cells into a pile. This individual child is not able to create a pile that's big enough to produce a grandchild xenobot. So this particular swarm gets a fitness value of 1.51. It gets 1 point for achieving 1 round of self-replication. And that child xenobot builds a pile which is 51% of the way to being big enough to start moving. So we tack on another 0.51. So in essence, what we're doing here is evolving kinematic self-replication or evolving replicative ability. Replicative ability in simulated xenobots is actually very difficult. If you just build xenobots by hand, not based on an evolutionary algorithm, they only replicate for 1 generation, and then that process runs out of gas. Mohammed dropped a question into chat, which is, are the parent xenobots homogeneous? Yes. So at the moment, we assume that all the nine initial xenobots have exactly the same 3D geometry and material distribution. Obviously, they can produce children that look and act differently from themselves. So Gus has a follow-up question here. Is there any equivalence of heredity here? That is a great question. The answer is we're not quite sure. It turns out that in certain conditions, a swarm will produce children that look and act more similar to themselves. In the video that you see here, the children obviously look nothing like their parents. We have absolutely no understanding of the conditions yet under which parents will produce children that look and act more like themselves, basically increasing the likelihood of heredity. Even if there is partial heredity in the system, it is obviously not normal heredity. There is no genome here. There is no genetic material. There is no von Neumann universal constructor tape being copied and pasted into the children. So we think, our group thinks that heredity is possible here, but again, it's not what you would normally expect heredity to be. Mohammed, go ahead, ask your question.
Hi Josh, it's very nice to see you. It's Mohammed, University of Southampton, UK. So I was wondering if through heterogeneity we could have some influence on the child or the children xenobots because of the way they interact in this world.
I'm sorry, Mohammed, I missed you at just the most important word. You said due to what?
So I asked if they are homogeneous. The reason why I asked is that if they were heterogeneous, we might have been able to think that the swarm could have some way of understanding how the children xenobot might look like because of the way they move in space.
Yeah, great point. So we are investigating now conditions under which you could increase heredity in the system, and heterogeneity in terms of 3D geometry, material property, and behavior within the swarm is one possible way that could be accomplished. Obviously, when you're dealing with 3D shape, a relatively easy way to get to heredity is complementarity. So you could imagine a convex xenobot pushing material into a concave xenobot that becomes a complement of one or both. That's one possibility. In the supplementary materials for this paper, Sam Kriegman, who did all the computational and evolutionary work here, actually evolved not the xenobot shapes, not the shapes of the xenobots, but the shape of the environment in which this process occurs. And it turns out that certain environments seem to have more heredity. The children look and act more similar to the parents in certain of these evolved environments than others, but it's just anecdotal. We were just sort of visually inspecting it after the fact. So I think this is still a wide open question, and if people are interested in this, we would definitely be interested in a collaboration and seeing if we can evolve heredity or somehow introduce heredity into the space system.
Thank you. Would love to, Josh.
Thank you, Mohammed. Great question. Okay, so just for those who hadn't seen it before, let me just finish this evolutionary trial here. So in this case, in this particular evolutionary run, we continued, and after a dozen or so generations, we obtained this particular swarm here, which the popular press were very happy to point out looked exactly like the Pacman character from the 1980s video game. Obviously, if you want to push stuff into large piles, it makes sense to have a concave leading edge and a convex trailing edge. So the Pacman shape, at least in retrospect, seems obvious, but none of us on the team were expecting that particular shape. Some of you may have experienced this in your own work, where people point out that something is blindingly obvious after you've reported the result. Maybe in retrospect it's obvious, wasn't obvious to us. So I would still count this as kind of a non-intuitive part of Robospace. The little schematic that you see just to the left of the video, I asked Sam to include this in the figure because I thought this was particularly interesting. You can see the 9 parents here lined up, and they collectively produce the 3 children that you see in the video, and those 3 children produce 1 grandchild. So in this evolutionary run, the swarm was able to postpone the loss of replicative ability for 2 generations. Turns out that 5 of the parents collectively built the pile that became the 2nd child. 1 parent produced the pile that became the 1st child, and so on. So you get differing contributions of construction or kinematic self-replication in the next generation. So this is another kind of interesting twist on heredity here. There's no reason why you have to have 1 parent or pairs of parents. You can have arbitrary number of parents that are collectively building a copy of themselves externally. Kind of an interesting thing, I think, to explore in future work. As this community probably knows, I don't even need to say it, one of the nice things about using an evolutionary algorithm is if you rewind the evolutionary tape and run it forward again, you often get diverse designs. Here are the end results of 49, I think it's 49, different evolutionary runs. There's a bit of convergent evolution here. You tend to get Pacman shape for obvious reasons, but still a fair bit of diversity here. I think I'm out of time. I'm just going to very briefly mention that we did one final computational study where we wanted to see whether this self-replicating swarm could perform useful work, which in this case was fixing a broken circuit as a side effect of replication. This is an important aspect of John von Neumann's original vision of kinematically self-replicating machines. Obviously, we don't want self-replication just for the sake of self-replication. Hopefully they do some useful work before they replicate out of control and destroy the planet. Okay. On that happy note, I think I will end. Here's some of my students and colleagues that contributed or did all of the actual work here that I described today. Thanks very much. If we have time, I'm happy to answer a few questions.
You bet we have time, at least I do, but I invite all others to ask the questions first. Please, feel free. Looking, no hands, except Karina Clipping. Thomas, go ahead.
I'm not saying. I'm not saying.
Hi, thanks very much for your interesting talk.
Sure.
I have a question about the latest experiment that you were discussing. Why is it that you are killing the parents? Why the parents cannot help producing further children?
Oh, great question. Yeah. So why are the parents not helping the children to produce grandchildren?
Yes.
I kind of jumped over the physical xenobots. So I'll just play the video of the physical xenobot parent making a child. Here's near towards the end. Here's a parent making a relatively large child. This child went on to make a grandchild. The xenobots at the moment, they quote unquote live for 7 to 10 days. There's still an argument ongoing on Twitter about whether xenobots are even alive or not. But after about 7 to 10 days, the ATP in the egg yolk that is stored inside these embryonic frog cells is all absorbed, and they run out of internal energy stores. So this parent will stop moving after about 7 to 10 days. It takes about a few hours or up to a day for the parent swarm to make the children. Takes another day or two for the children to mature and grow these cilia. So by the time the children are moving, the parents are reaching the end of their lifetime. There is still, however, significant overlap. So I think you're right. We could, in theory, leave the parents in the dish, and we could study grandparent scaffolding of kinematic self-replication. We didn't here. We separated the generations, but I think you wouldn't have to make any change to our experimental setup with the physical xenobots to test questions about that. Yeah.
Okay, thank you.
Great question. Thank you.
Okay, all others, don't feel shy. This is your chance. Mohammed, go ahead.
Sorry, I'm trying to seize this opportunity and no one else is asking question. So Josh, where do you see this going? Are there some medical applications or anything you have in mind?
Yeah, lots of different directions. On the engineering side, we are building some biofabrication facilities so that we can automate the manufacture of these xenobots. We obviously need to get away from artisanal robotics. Even if we ask the xenobots to make more xenobots, that's also still a relatively slow process and an unreliable process. So a lot of work on biomanufacturing. You mentioned about applications. So we have a spin-off company now called Fauna Systems, as in flora and fauna. And we're in discussions with a lot of different industrial domains. We mentioned in the first paper, kind of offhand, environmental remediation, finding microplastics and removing them from waterways. That may be possible in the long term. In the short term, actually, some of these medical applications might become possible. We're looking at trying to build xenobots or biobots from cells taken from other species. It may become possible in the future that we could build biobots from human cells. Might be possible to build biobots from a human patient's own cells, in which case if they ingested some of these robots to do internal work inside the body, you could sidestep the patient's immune response. That's obviously a long way off. There would be huge regulatory hurdles for good reason. So I think practical applications are still quite a ways off. In the shorter term, I think what's interesting, what we're pursuing is actually creating fitness functions for our evolutionary algorithms that reward xenobots not for walking or swimming or replicating, but for being conceptually transparent. Meaning that they advertise their internal function as they behave better than naturally evolved organisms do. Obviously, for good reasons, naturally evolved organisms hide what's happening inside from the outside world. We can perhaps create xenobots that allow us to better unlock how cells talk to one another, how cells change how they talk to one another when they're rearranged in phenotypes different from default phenotype. So I think it's going to be interesting to explore xenobots as kind of a new biological tool in the next few years.
Okay. Thank you, Josh. Thomas or someone in the neighborhood of Thomas has a question.
Yeah, I'm afraid it's again Anya that has a question. It might be a silly question, but I'm still going to ask it. From the experimental point of view, have you ever tried running experiments in 3 dimensions?
Yeah, great question. We are working on that, and I can't say too much about it, obviously, because it hasn't been published yet. The short answer is yes, you can create xenobots that move in 3 dimensions.
And they are effective also in 3 dimensions.
I'm sorry?
And they are effective also in 3 dimensions.
It depends on what you mean by effective. At the moment, they can navigate. They can move in 3 dimensions. I forgot to mention it has to be room temperature fresh water because obviously these are frog cells. So obviously the environmental niche for these micro robots is still somewhat limited to the environmental niche of their natural donors.
Okay, thank you.
Yeah, thank you.
So I raise my own hands just to encourage the others. Self-replication, obviously there are very dystopic scenarios based on that word, which brings me to the question, have you thought of a kill switch? How to shut down the system?
Yeah, good. The kill switch, right? The answer is there is never a 100% reliable kill switch. What we're hoping for is that this provides a novel scientific tool for studying self-replication. The reason we're all here in Zoom and not in person is because obviously as humans, we still grapple with controlling biological systems that self-replicate out of control. We don't understand how that occurs or the conditions under which that occurs very well yet. First of all, the xenobots are very safe. These are genetically unmodified frog cells. Frogs shed untold trillions of cells into freshwater ponds all day, every day. So even if these things escaped from the dish, we wouldn't be in danger of the gray goo scenario. So it is a very, very safe system. in which to explore conditions under which we can amplify or suppress or channel self-replication. I hear you about the kill switch, but I think when it comes to self-replication, we have to think carefully. Actually, one of the most dangerous things you can try and do is build in a kill switch, because then evolution will do whatever it can to sidestep it.
Yes,
I think it's yep.
Sorry, go ahead.
I was just going to say, one of the things we explored in this paper is all different kinds of fitness functions that select for different kinds of replication. Speed it up, slow it down, produce bigger offspring, smaller offspring. We're never going to be able to suppress self-replication in the natural world completely. All we can do is try and channel it as best we can, and I think this is a scientific toolkit for studying those kinds of questions.
So one more recommendation. Probably you have heard about Alex Ellery, but if you haven't, then I want to name. Do you know him or his work?
I sent him our manuscript the day it was published, because Alex and I have spoken about these ideas for a long time. Yep.
Good. Okay. Yeah. Thank you. So giving back the floor to the others in the audience. So Josh is with us for more than an hour, entertaining us and educating us. So I'm willing to give him a break now, so you have a few seconds to raise your hand and ask your question. Rien ne va plus. Okay. Thank you, everyone. Thank you, Josh. It was really a pleasure. It was really great seeing you. Thank you very much for the lecture, and looking forward to see you in real life.
Absolutely.
Awesome.
Thank you so much, everyone. It was nice to visit with you today.